We are going to go ahead and get started this morning. Um, uh, uh, I'm Dr. Jennifer Trainer, and I'm introducing this morning, my pleasure to introduce Daniel Leeper. He's a third-year pediatric resident here at Lurie Children's, and Dr. Shanley asked me specifically to convey how excited he is that one of our residents is presenting today at Grand Rounds, which we think might be the inaugural appearance of a resident presenting at Grand Rounds. Certainly in the 26 years since I was a resident here, we haven't had one present. He also wanted to convey how sorry he is that he's not able to be here in person to introduce you. Many of you know Dan, but what you might not know is that Dan is passionate not only about clinical medicine, but also about biosecurity, the field that focuses on combating emerging threats to health. Dan's path to pediatric residency was probably a little more unusual than most. During his undergraduate years at Stanford, he studied international security at Stanford University's Center for International Security and Cooperation and worked in the Science Advisory Office of the U.S. Department of State as their lead representative on pandemic flu during the 2009 H1N1 swine flu pandemic. Dan completed his undergraduate medical education at UC Davis, where he graduated and was inducted into AOA, and also earned the Public Health Sciences Departmental Award. During medical school, Dan rotated in the influenza division of the Centers for Disease Control, and most recently, during residency, he completed a part-time fellowship in biosecurity at the Johns Hopkins Center for Health Security. These experiences have deepened his passion for the topic today, which is genome editing, its promise, and its perils. Please join me in welcoming Dan.
All right. Thanks, Dr. Trainer, and good morning, everyone. I have no conflicts of interest to disclose. I'm honored that today I get to speak about biosecurity, which is the field that deals with protecting against threats to health. Today, I will discuss genome editing. Our ability to manipulate genes and edit the human genome has advanced incredibly quickly in recent years. So what is so important about gene modification? Well, most diseases we encounter in pediatrics are either caused by or influenced by a patient's genes. The ability to modify genes, coupled with a good understanding of genetics, would let us prevent or cure many of the diseases that afflict our patients. Gene modification will increasingly impact the field of pediatrics over the course of our careers. My talk today has 3 parts outlined here in question form. First, I will explain some of the recent advances in the genetic tools that allow for gene modification, especially DNA sequencing and the CRISPR toolbox of gene editing. I also will discuss a powerful outgrowth of CRISPR, which is called gene drive. Second, I will discuss what is so promising about our new ability to sequence DNA to manipulate genes and give examples of ways that they are beginning to revolutionize the field of pediatrics. Lastly, I will discuss my particular research interest, which is how advances in gene editing technology might run us into a good deal of trouble. I want to illustrate some of the possible unintended consequences of this revolution in gene editing. In the past few years, there have been dramatic advances in our ability to sequence DNA, synthesize new genes, and to manipulate the human genome. Let me introduce you to some of the advances in genetics, and I'll start with DNA sequencing. Understanding genetics and modifying the genome requires data, and obtaining these data requires DNA sequencing. Whereas DNA sequencing used to be incredibly expensive, the cost has plummeted, and the reason is that our methods of sequencing DNA have advanced considerably. One example of a recently developed, high-throughput DNA sequencing method is nanopore sequencing. In it, strands of DNA or RNA are threaded through a nano-sized pore. Each nucleotide gives off a unique electromagnetic signal as it passes through the nanopore, which is read in real time as the oligonucleotide passes through. This method has several advantages over older methods, such as the Sanger method. It leaves DNA intact instead of destroying it. Since the method does not rely on PCR like prior methods, nanopore sequencing does not require the use of DNA primers and can also be performed over a wide range of temperatures. This means that nanopore sequencing can be performed outside of the controlled environment of the laboratory. For $1,000, you can buy a nanopore DNA sequencer that fits in the palm of your hand. Nanopore sequencing is just one of the many high-throughput DNA sequencing methods that are in use today, and I've listed some common methods here. There's no single best sequencing method, and which method is most suitable depends on the situation. The development of new DNA sequencing methods has driven down the cost of DNA sequencing. This is a chart from the NIH showing just how tremendous of a drop it has been, and note that the y-axis here is on a logarithmic scale. In 2007, it cost about $10 million to sequence a human genome. 8 years later, the cost was about $1,000. That is a 10,000th of the cost. And it's astounding that if you think of a Toyota Camry, and imagine if in 8 years a Camry cost only $2.31. That is how quickly the cost of DNA sequencing has gone down. So take a look now at the white line on the chart. The wording is pretty small there, but it says Moore's Law. So this line represents Moore's Law, and it's a concept from computer manufacturing. Gordon Moore co-founded Intel, and in the 70s he observed that every 2 years computer chip manufacturers were able to fit twice as many transistors onto a dense integrated computer circuit. He correctly predicted that this trend would continue, and he defined this rate of progress as Moore's Law. Moore's Law predicted that computing power would increase exponentially, while costs would decrease exponentially. And just as Moore predicted, computers have gone from being huge pieces of equipment that occupy entire rooms to now being compact machines that can fit in your wristwatch. The iPhone that you carry in your pocket has far more computing power than all of NASA possessed when they landed astronauts on the moon. Put another way, computers have proliferated in that there are many more computers in many more contexts than ever before. Computer technology has also become democratized, meaning that the technology has become broadly accessible with low barriers to entry that permit more people than ever to participate in the development of new computer products and computer applications. So if we look again at the chart, you will observe that the drop in the price of DNA sequencing is significantly outpacing Moore's Law. Think of how revolutionary the change in computing power has been for our society. This genetic revolution is moving faster than Moore's Law, and it doesn't involve silicon, it involves DNA, the building blocks of all life. Because of this drop in price, DNA sequencing is proliferating, and it is becoming democratized. As a result, DNA sequencing is quickly cropping up in places that you may not have expected. For example, an apartment building in the West Loop here in Chicago performs DNA testing on dog poop to identify which owners here have not been cleaning up after their pets. In another example, in 2013, Irish investigators tested hamburger meat that was being sold in Irish supermarkets and found a significant amount of horse DNA instead of cow DNA. A subsequent investigation uncovered that much of the European Union's beef supply chain was adulterated with horse meat. Here in the US, investigators using DNA analysis have found widespread mislabeling of fish. DNA sequencing also has a growing number of uses in the clinical setting, which I will discuss later on. It is not just the price of sequencing DNA that has dropped, which on this graph is shown in blue. The price of synthesizing DNA, which is shown here in the red and yellow, has gone down as well. The size of the machines has shrunk too. This is a commercially available DNA synthesizer that fits on a desk. So our ability to sequence DNA and synthesize DNA has advanced by leaps and bounds. How about our ability to modify the genetic makeup of organisms? This too has taken a quantum leap in the form of the CRISPR toolkit. CRISPR is a new gene editing method that has been developed within the last 10 years. Compared to previous methods of genetic modification, it is extraordinarily cheaper, simpler, more reliable, and more precise. I'll give a brief history of genetic modification. we have been modifying the genes of the organisms around us for millennia. The earliest form of genetic modification is selective breeding, where animals with certain traits are selected out and preferentially bred. And so thanks to selective breeding, you can put all kinds of cool goldfish in an aquarium. Selective breeding is what gave us domesticated animals like dogs and cows. But selective breeding is limited to the natural variety found within a species. Decades ago, scientists developed a tool called recombinant DNA that made it possible to introduce DNA from one species into another using phages. With recombinant DNA, we can add even more exotic creatures to our aquarium. So this is a jellyfish that has a protein that makes it fluoresce green. So say you took that gene, which is called green fluorescent protein, or GFP, and then spliced it into a zebrafish. The result is a glowfish. Glowfish have been on the market for over a decade, and they now come in many colors. But recombinant DNA still has its limits. It is cumbersome and is largely limited to the genetic variety found in the natural world. Newer methods of modifying genes were developed in recent years, including retroviruses, zinc finger nucleases, and talins. These methods have all been largely superseded by the CRISPR toolkit, which represents a quantum leap in our ability to modify genes because of its precision, its ease of use, and its low cost. Understanding CRISPR is going to help you with understanding the rest of my talk. CRISPR edits DNA in much the same way that Microsoft Word's search and replace function edits Word documents. CRISPR lets you search for a certain stretch of DNA, excise it, and then insert a new custom stretch. The CRISPR toolkit is derived from genes that are found naturally in some bacteria and archaea. A protein, most commonly one called Cas9, binds a sequence of RNA that is engineered to match a sequence of DNA in a genome. When the Cas9 RNA complex binds to the complementary stretch of DNA, Cas9 cleaves it and allows for another custom sequence of DNA to be swapped in. This is the CRISPR-Cas system, and you'll notice that the 9 is not pronounced. CRISPR is encoded by DNA, so introducing CRISPR into a cell turns out to be very straightforward. For example, CRISPR can be encoded on a plasmid that's introduced into a cell. The CRISPR method is constantly being refined, and the CRISPR-Cas system is being superseded by newer, more accurate CRISPR-based genome editing methods. For example, new forms of CRISPR localize to a target stretch of DNA and then modify a single base pair without ever cleaving the DNA. Another derivative of CRISPR is called no-cut CRISPR. Here, a CRISPR binds upstream of a gene and is affixed to a transcriptional activation domain. The net result here is that CRISPR acts as a gene promoter, increasing transcription of a selected gene without ever modifying it. CRISPR is fairly inexpensive, and simple kits are available for purchase online for about $100. Here's one that I found on Amazon, which even comes with its own pipette. So while I'm shopping online, after I bought my CRISPR kit, where could I just buy some genetically altered stem cells? This is a screenshot of Harvard University's Harvard Stem Cell Institute. They have a whole catalog of genome editing services for purchase. They sell human pluripotent stem cells to the private and public sectors. If you work for an academic institution, you get a discount, and for under $18,000, you can order a line of human pluripotent stem cells with a gene knockout of your choice using CRISPR. Alternatively, you can order a line of human pluripotent stem cells that have point mutations that are customized to your liking. Let me give an example of how powerful CRISPR can be by telling you about a recently developed genetic method called gene drive, also known as mutagenic chain reaction. Gene drive subverts Mendelian inheritance. I will walk you through an experiment done by a team at UC San Diego that explains what I mean by that. In fruit flies, there is a single gene responsible for pigmentation. If a fruit fly has 1 or 2 normal or wild type copies of the pigment gene, it expresses normal pigment, like this one. If a fruit fly has 2 recessive copies of the pigment gene, it is albino. What the team at UC San Diego did was mutate a fruit fly's genome so that it expressed a Cas9 protein that was targeted to find normal copies of the pigment gene and swap in instead the mutant albino copies of the gene. Now, they took the mutant fruit fly and then crossed it with a normal wild-type fruit fly. Now, if this were Mendelian inheritance, here's what we would expect. We would expect all of the progeny to be heterozygotes because they would have gotten one mutant copy of the gene from the mutant parent and one wild-type copy of the gene from the wild-type parent. But gene drive subverts Mendelian inheritance. And so in this experiment, 97% of the offspring were albino. Here, gene drive prevented Mendelian inheritance, making the offspring inherit 2 mutant copies of the gene from the mutant parent and 0 copies of the wild-type gene from the wild-type parent. And when these mutant albino offspring mate with normal flies, their offspring are mutants as well, and so on. Gene drive forces a mutation into a population, eradicating normal copies of the gene. And this is why gene drive is also known as mutagenic chain reaction, because introducing a single mutant sets off a chain reaction of mutations in the entire population. All of these advances in DNA sequencing, DNA synthesis, and genome editing have now gotten us to the point where we are technologically able to edit the human germline. Let me explain what I mean by that. Germline cells are the set of an organism's cells that are capable of passing their genetic material on to their offspring. Somatic cells are the name that we give to the set of cells that are not capable of being passed down to offspring. Our current gene therapies only alter the genomes of somatic cells. For example, here in this hospital, when we perform stem cell transplants on patients with, say, sickle cell anemia, we ablate their bone marrow cells and replace them with myelocytes that no longer carry the sickle cell genotype. We've modified the somatic cells, but the patient's germline cells still carry the original sickle cell genotype. Now, granted, stem cell transplants do render most patients sterile, but if a patient could still reproduce after their stem cell transplant, that patient's offspring would still receive the sickle cell mutation. CRISPR, though, makes it technologically and financially feasible to genetically alter human germline cells. The way this is done is by modifying the human embryo while it is just one cell, or by genetically modifying gametes, such as the egg, prior to fertilization. In this way, genetic changes induced by CRISPR make it both into the somatic and the germline cells. Now, historically, there has been an ethical barrier to modifying germline cells because it means not just changing a person's genes, but the genes of future generations. Today's experimenters have gone right up against this ethical red line by successfully editing human embryos, but not carrying them to term. They discard them. And so again, germline editing of a human embryo culminating in pregnancy is widely considered to be unethical. That said, because this technology is barely regulated, any day now, news might break that a research team has crossed that ethical red line by editing the germline of human embryos and then bringing those embryos to term. In the United States, doing so appears to be perfectly legal. To summarize this section of my talk today, there have been leaps and bounds in our ability to sequence DNA and to synthesize DNA nucleotides. The cost of these technologies is plummeting, far outpacing the explosive rate at which computing power developed in recent decades. Now we have a new powerful gene editing tool called CRISPR that makes gene editing cheaper, easier, and more reliable than ever before. There also is an outgrowth of CRISPR called gene drive, which forces all offspring to receive only mutant copies of a gene and prevents them from receiving any normal copies of the gene. Lastly, early efforts at editing the genomes of human embryos have been successful, and the ethical red line that prohibits editing of the human germline might be crossed any day. Now I shall move on to the second portion of my talk, which is to explore what is good about these technological advances in genetics. How might they be used to improve the health of children? I will circle back to the technologies I described in the first part of the talk to give a few examples of how they're being used now to alleviate disease and to peer into the near future at some emerging technologies. Let's revisit our ability to sequence DNA more cheaply than ever. I'll give examples of some specialties that are benefiting from inexpensive DNA sequencing. I'll start with infectious diseases. Advances in DNA detection have already changed the way we detect infectious diseases, with genetic-based assays now replacing culture-based methods. In this hospital, we sequence bacterial genomes to predict antibiotic sensitivities. Now that sequencers are smaller and easier to use, we might increasingly perform DNA tests at the point of care instead of needing to send them to the lab. As DNA detection becomes cheaper, there may be a paradigm shift from diagnosis to surveillance. Currently, we diagnose patients once they develop clinical symptoms like fever and cough. But with inexpensive sequencing, we gain the ability to identify infections in the pre-symptomatic stage, just as we identify metabolic diseases in newborns before they've ever become clinically apparent. This potentially gives us the ability to treat an infection before it ever takes hold, and potentially before it gets transmitted to someone else. Currently, epidemiologists monitor the sewage in countries around the world to test for clinically inapparent polio that's excreted in the stool. Perhaps in the future, we will have DNA sequencers in our homes to passively monitor our saliva, urine, and stool for pathogens. Sequencing also helps with infection control in the hospital. Since viruses mutate as they hop from person to person, some hospitals have managed to trace the path of transmission of nosocomial infections by sequencing the bugs found from each patient who comes down with it. Next is neonatology. Amniocentesis is becoming increasingly replaced by cell-free DNA testing, which involves analyzing the fetal DNA that's circulating in the maternal blood. Because the costs of DNA sequencing are plummeting, it may soon be feasible to sequence a fetus's entire genome by sampling the pregnant mother's blood. Talk about the field of genetics. As sequencing becomes cheaper, it is relatively easier to sequence a patient's whole exome or whole genome. Although it does cost some to interpret the genome, right now it costs about $1,000 to sequence a human genome. When genome sequence data are pulled from a large population level studies called genome-wide association studies, or GWAS, can elucidate how certain genes contribute to particular diseases. As we sequence more patients, our therapies could become increasingly personalized, just as how now in cystic fibrosis and in epilepsy, there are treatments that are targeted to the particular genetic mutation that a patient possesses. This approach of targeting therapies to an individual's genetic makeup is called personalized medicine. Next is oncology. With inexpensive sequencing, it would be easier to check the blood of patients who have been treated for leukemia to see if there's evidence of recurrence. Separately, in solid tumors, small quantities of tumor DNA circulate in the blood. If this circulating tumor DNA were sampled and sequenced, it might serve as a minimally invasive liquid biopsy that could someday substitute for obtaining a tissue sample. And as one last example, gastroenterology, it's increasingly apparent that the composition of the gut microflora contributes to conditions such as recurrent C. diff and obesity. By sequencing gut microbes, we can better understand the interaction between gut microbiota and disease, and perhaps identify which compositions of gut microflora warrant therapies like fecal transplantation. Okay, so now let's switch gears to gene editing. Again, I will give an overview of just a few examples of the ways that gene editing is already advancing or may soon advance fields related to pediatrics. As we go along, please keep your eye on the publication dates of the papers I cite, and you'll see just how recent some of these advances are. I'll start with hematology and oncology. At our hospital, we've begun using CAR-T therapy to treat refractory B-cell lymphoma. How this works is that T cells are removed from the blood and genetically modified using a retrovirus. These genetically modified T cells express a receptor that's targeted to surface proteins that are expressed by B cells. In this way, the body's immune response is directed against cancerous B cells, and early results have been promising. Next up is pediatric dermatology. There is a severe skin condition known as epidermolysis bullosa, or EB, where the skin blisters off and sloughs off easily. In severe forms, it's often fatal. A team in Italy successfully treated a young patient with severe EB through genetic modification. They harvested stem cells from the patient's dermis and then used a retrovirus to genetically correct the mutation in those stem cells. These genetically modified cells were then grown in sheets and then grafted back onto the patient. And since it's the patient's own skin, there's not a concern for immune rejection. You can see here the dramatic improvement. My next topic is drug manufacturing. Insulin used to be harvested from pancreases of cows, pigs, and fish, but since the 1980s, it has been manufactured using genetically altered yeast. Advances in genetic engineering make it possible to manufacture a wider variety of medications using simple organisms like yeast. For example, teams at MIT and Stanford genetically engineered a yeast that converts sugar into morphine. If opiates were made by yeast, we wouldn't need to harvest poppies from abroad. Also, if people got their hands on these yeast, they could homebrew opiates in their bathtubs. Next is organ transplantation. If organs could be transplanted into humans, it might solve our perpetual organ shortage. Well, pigs are a tantalizing source of organs since they are relatively human-sized and relatively easy to cultivate. Scientists have raced to use CRISPR to genetically knock out porcine-specific antigens and retroviruses that elicit immune responses when the porcine tissue is engrafted into other animals. Last year, a team at Emory announced that they had taken a kidney from a genetically altered pig, transplanted it into a rhesus macaque monkey, and it lasted more than 400 days before being rejected, a new record. But perhaps instead of transplanting animal organs into humans, we could have animals simply grow human organs. A team at the Salk Institute announced that it had implanted human brain stem cells into the visual cortex of a mouse brain, giving rise to human neural tissue that demonstrated neural activity. This brings up an ethical issue: how much of a mouse brain can be made up of human brain cells before we have to start treating that mouse as partly human? A different team at the Salk Institute published a paper last year showing that they had formed human-pig chimeras. Human stem cells were injected into an early pig embryo, giving rise to a pig embryo in which about 1 in 10,000 cells were human cells. This picture here shows human cells, which fluoresce green on the right, in a sample of tissue from the pig embryo. The experimenters aborted the embryo after 28 days. Now that we've spoken about ways that genome editing is being incorporated into the field of pediatrics, I would like to revisit gene drive. Gene drive turns out to be a potential magic bullet for stopping vector-borne infectious diseases such as malaria and Zika. As it turns out, female mosquitoes bite humans and males do not. If male mosquitoes were introduced into the wild with a gene drive that made female progeny non-viable or sterile, a massive gender imbalance would develop. The outcome would be a mass die-off of that mosquito species, potentially driving it to extinction. The Gates Foundation is investing heavily in gene drive, and open field experiments on gene drives in mosquitoes are in the works in several countries, including the United States. You can see now the tremendous potential of DNA sequencing and genome editing. DNA sequencing gives us new ways to detect and monitor diseases, and gene drive allows us to manipulate the genomes of entire populations. So while genome editing has the potential to cure a number of diseases, it also is frighteningly dangerous. In April 2016, General James Clapper, who at that time was the U.S. Director of National Intelligence, testified to the Senate Armed Services Committee that genome editing had become a global danger and a threat to national security. He classified it as a weapon of mass destruction, on par with nuclear and chemical weapons. He is not the only one within the Intelligence Committee who is worried. As part of my biosecurity fellowship this last year, we met with intelligence agents at the CIA, the FBI, and the Pentagon who expressed alarm at the destructive potential of these technologies. And so I want to transition to the third part of my presentation, which is to explore ways that these new genome editing and DNA sequencing technologies could hurt us tremendously. I would categorize the risks in 3 broad categories: intentional misuse, accidental release, and unanticipated consequences. In discussing these three categories, I touch upon a few of the many privacy and ethical concerns that are inherent to gene editing. First is intentional misuse. I discussed earlier that when the price of a technology plummets, it tends to proliferate and become democratized. Already, we can see how gene editing is becoming democratized. There is iGEM, which is an international science fair for students that involves genetically altering cellular organisms. In some cities, you can find biohacking labs where you can rent wet bench space and laboratory equipment by the hour and perform genome editing experiments. So what if someone wanted to manufacture a pathogen? Pathogens are based upon nucleic acids, and it follows that as it's easier and cheaper to synthesize DNA and RNA, and easier to get a hold of the equipment to do it, it becomes easier to synthesize pathogens. Poliovirus turns out to be quite easy to synthesize, partly because it has few genes and a very short genome. In 2002, which is ages ago compared to these other studies that I've been citing, an American team successfully synthesized live poliovirus in a single test tube. So that's just poliovirus, and at least we're not talking about synthesizing something truly scary like smallpox, right? Well, last year, a Canadian lab recreated the horsepox virus, which is an extinct pox virus that is an extremely close relative of smallpox. They did it using DNA sequencing that they ordered online and received in the mail. A WHO panel noted that it did not take exceptional biochemical knowledge or skills, significant funds, or significant time for the researchers to recreate horsepox. Whatever technical steps are required to generate horsepox are the same technical steps required to manufacture smallpox. You don't even need a sample of live smallpox in order to obtain a sequence. In fact, the genetic sequence of smallpox is available online for free on the NIH website. And if you'd like to see, I've put some of it right here. The 1918 flu infected a third of all people in the world. Its full genetic sequence was published in 2005. And since then, teams have indeed recreated active virus from that sequence. In fact, several labs in the US have conducted what is known as gain of function research on influenza, SARS, and MERS, where they intentionally genetically alter these viruses to make them more virulent and more lethal than existing strains. In response to an outcry by some members of the scientific community, the NIH did decide in 2014 to temporarily suspend its funding of gain of function research. Last month, it lifted the moratorium. As we democratize the ability to synthesize genes, we give more people the ability to manufacture deadly pathogens. And unlike a nuclear weapons program or a chemical weapons program, you don't need secret laboratories and smuggled equipment and sponsorship by a nation state. You just need some lab equipment and some scientific know-how. There also is the possibility of synthesizing not just known or extinct viruses, but entirely new viruses that have never been seen before. Remember too that I discussed personalized medicine earlier, which involves tailoring therapies such as medications to an individual's genetic makeup. The technologies behind personalized medicine would also allow an actor to tailor a poison to an individual's genetic makeup or to target a pathogen to a particular ethnicity. Once again, we're seeing this phenomenon where the good technology and the bad technology are two sides of the same coin. There is a potential here for targeted killing on a massive scale. And how about gene drive? Well, gene drive would be an extraordinary biowarfare agent, again because of its precision. Let's say that an actor wanted to attack the US and only the US, and it used gene drive. It could target a crop that is grown almost exclusively in the United States, like wheat. A few wheat seeds could be modified to carry a gene drive that would force expression of, say, a neurotoxin. By planting a few of these genetically altered wheat seeds, the toxin gene would insert itself into wheat crops all around the country within years. This type of attack would be inexpensive to carry out, it would pose little risk to the actor, it would be difficult to detect, and once discovered, it would be difficult to fix or to even figure it out who carried out the attack. To me, it makes sense why General Clapper was so alarmed. The risks of these technologies are not just limited to those who intend to harm. There also is a chance of accidental release of harmful agents. Sometimes experiments don't stay in their cage. Killer bees were the result of a breeding experiment, and in 1957 they escaped their enclosure and spread across the Western Hemisphere. So now think back to the gain-of-function influenza experiments that I described a few minutes ago, where researchers in the US intentionally mutated strains of influenza to become more virulent, more lethal, and more resistant to existing vaccinations. Could this lethal flu virus escape the lab by mistake? Well, history tells us that accidental lab releases do occur. Genetic analysis shows that a global outbreak of H1N1 flu in 1977 was apparently due to a laboratory's accidental release of a stored 1957 flu strain. There have also been recent high-profile mistakes in the US. In the year 2014, vials of live smallpox virus were found in a storage room in the NIH. In that year, the CDC mistakenly sent a sample of highly pathogenic avian flu to a bird research facility, and 80 CDC workers were exposed to weaponized anthrax. If accidental releases occur at world-class institutions like the CDC and the NIH, who is to say that accidental release wouldn't occur at private sector facilities where there might be a financial incentive to cut corners, or in countries whose laboratory safety standards might not be as stringent as ours? And what if nature itself is being experimented on? The Gates Foundation is planning gene drive experiments in Mali, Uganda, and Burkina Faso, experiments that will involve releasing mosquitoes with gene drives directly into the wild. Once we have caused a mutagenic chain reaction in the wild, how can we possibly hit the reset button? These genetic changes to the population cannot be reversed. And in this last part of this section about the risks of genome editing technologies, I want to discuss possible unanticipated consequences. As far as genome sequencing, privacy issues abound. We shed genetic material constantly, including when we shed hair or when we drink from a cup. In the US, generally, our DNA is no longer our property once it has left our bodies, meaning our DNA is fair game to be sequenced by whoever gets their hands on it. Law enforcement is using genetic sequencing in new ways. As an example, over a dozen crime investigations in the US have used a method called familial searching. For criminal cases where the suspect's DNA doesn't have a match in a database, investigators query the database to find genetically similar DNA. When they find partial matches, they can infer how closely related that match is to the suspect. And with some math and some genealogy, they can deduce the suspect's identity by looking at partial matches in the database. Privacy advocates have pointed out that this approach discriminates against minorities and poor people, since those are the people who are most likely to already be in law enforcement DNA databases. Let's move on to genome editing. One issue is that it's not clear which genome edits are ethical and which ones are not. This is a photo of Eero Montaranta. He had a mutation of his erythropoietin receptor that caused an increase in the oxygen carrying capacity of his blood. He put this, and also it's important to note too that this mutation did not seem to cause any ill effect. He put this mutation to good use. He competed as a cross-country skier for Finland, and he won 7 medals in the Winter Olympics. So my question is, if we're going to edit someone's genome anyway and eliminate their Huntington's disease or their Gaucher's disease, why not just include this mutation too and make them an Olympic athlete? If we're going to introduce a beneficial mutation to increase the oxygen carrying capacity of the blood, why not also include known beneficial mutations that decrease the likelihood of obesity, of Alzheimer's disease, or of HIV infection? And while we're at it, if we're already doing that, why not make them taller or modify their eye color or lighten the color of their skin? There is no clear boundary between removing a disease and adding an enhancement. And here once more, we have a situation where the good and the bad are two sides of the same coin. Human genome editing methods that remove disease are the same ones that are used to create designer babies or to genetically enhance living people. Now, you might respond that I'm being speculative and that people biohacking their own DNA is something in the distant future. Well, there are already reports of researchers and entrepreneurs using CRISPR to modify their own cells. And you'll remember the green fluorescent protein I told you about that comes from a jellyfish and that makes a glowfish fluoresce. Let me introduce you to Josiah Zahner. He's a biochemist who got his PhD at UChicago. A few months ago, he injected his skin cells with a CRISPR modification to make them express green fluorescent protein, and now some of his skin cells do fluoresce green. More recently, he injected his muscles with a CRISPR modification in an attempt to make his muscles larger. He is part of a small community of biohackers who are injecting themselves with CRISPR, and at the moment, what they are doing is perfectly legal under US law. So how might this democratization of genome editing technology play out internationally? Remember that Russia went to extreme lengths to dope its athletes in the 2014 Sochi Olympics and cover up the evidence. So my question is, if Uzziah Zaner is injecting his muscles to make them bigger, why wouldn't Russia want to attempt the same in its athletes? Because also, this method would escape detection under conventional drug detection methods. And for that matter, why don't countries around the world want to modify the genomes of their soldiers or of their newborns? Genes are powerful, and I can't see why countries wouldn't want to use genome editing to get an edge. It's possible that they're doing so already. One last concern they'll bring up is that these technologies could exacerbate inequality. To carry out genome editing on human embryos, we need to implant the embryos back into the mother using IVF. IVF is quite expensive. And so who's genome editing going to be available to? Is it going to be available only to wealthy countries, only to wealthy people? As these technological advances have continued at pace, the national and international regulatory framework that regulates biosafety has not kept up. For example, Canada has laws against germline human genome editing, but the US does not. The US government agencies that would advise on these issues and collaborate with other countries have been depleted of senior staff. Many crucial positions remain unappointed, and I've listed some here on these 3 slides. As some examples, the White House Office of Science and Technology Policy, also known as OSTP, has no director and has no associate directors. The Department of Homeland Security has no undersecretary for science and technology. The State Department has no science and technology advisor, which is the office I used to work in. 60% of the State Department's top-ranking career diplomats have been fired or have left. Worsening the situation, we've antagonized many of the countries that we would need to collaborate with, and the current administration's decision to withdraw from the Paris Climate Accords has diminished our international standing on scientific matters. In the absence of international leadership, the international framework to regulate these dangerous new technologies is weak to non-existent. The U.S. National Academies issued a report in 2016 and another in 2017 that largely gave the green light to gene drive and human genome editing experiments. Some of their justification was that, ethical concerns aside, if we don't perform the research here, other countries will beat us to it. These technologies are currently developing in a regulatory vacuum, and that makes it so it's not clear where all of this is going to lead. To conclude my Grand Rounds presentation, I would like to highlight some conclusions from my talk. One is that we are much farther along than many of us probably realized as far as our ability to edit the human genome. The era of custom genomes and designer babies is nearly upon us. Second is that this gene editing is double-edged because the same technologies that give us the ability to combat, prevent, and cure diseases can also be used for nefarious means. And a third is that these technologies have advanced faster than our ability to regulate them, which potentially opens us up to problems down the line. So what should we, as pediatricians, make of these double-edged technologies? I firmly believe that these genetic technologies will increasingly impact us as pediatricians. We're already employing some of them in the clinical setting. Also, if regulations are not put into place, within years, we might be caring for babies whose genomes have undergone elective modifications. And soon, we could also find ourselves dealing with a pathogen that was created in a laboratory. But we are able to influence the future. And so today, we as pediatricians have a choice. Do we remain on the sidelines, or do we choose to engage with this genetic revolution? Should we choose to engage, I think we're in a good position to do so because we occupy a position of public trust. The public trust that we act in the interests of children and that we strive to balance the needs of today's patients with the needs of future generations. If we wanted to, we could become a vocal counterweight to the rush to adopt human genome editing and gene drive by calling for and even helping to craft new regulations. If we as a specialty wanted a place to start, we could encourage the AAP to issue a policy statement on germline editing or on gene drives, as the genetics specialty has already done. But if we are going to act, the time that we would make the most impact is right now, while these technologies are still in their infancy and while there still aren't that many players involved. If we delay, I'm convinced that we will lose our chance at having a seat at the table and more cats are going to be let out of more bags. For better or worse, this genetic revolution is real and human genome modification is happening now. My hope is that we will engage, and I hope that we will manage to successfully strike a balance to realize the benefits of gene editing while avoiding its calamities. In closing, I want to thank the program leadership, especially Dr. Sharon Unte, for allowing me to pursue my interests in biosecurity during residency. I want to thank Dr. Jen Trainer for inviting me to give grain rounds today and for her introduction, and also to the faculty who helped me refine today's presentation. I want to acknowledge my co-residents on this side, the chief residents, and my faculty mentors. I owe gratitude to Stanford's Center for International Security and Cooperation, also called CSAC, to the Johns Hopkins Center for Health Security, and to my medical school mentor, Dr. Nathan Fairman, for helping me to pursue my interest in this subject. I'm also grateful to my family for supporting me throughout. Now I will open up the floor to your questions, and I appreciate you all attending.
Thank you very much, Dan. That was an inspiring and terrifying presentation. And I have definitely been rewarded for taking a chance and letting you present at Grand Round. So thank you very much. You will be a hard act to follow after the inaugural president presentation.
We had no doubt that this was going to be incredibly successful. Does anyone have questions?
Or comments?
Maybe just one comment. First of all, that was terrific. So, you know, in the 70s, when DNA recombination technology first came out, people were just very scared of it, right? It took people a while. I mean, they thought this was going to be, you know, and so a lot of these regulations came into play. I'm not even sure they made all that much sense. I mean, for example, they clearly made the difference between germline and somatic just because people were so afraid of it. I mean, you know, I think that as an Aristotelian, you need to be able to take the middle road. There's a very difference, you know, between taking out the sickle cell anemia gene or taking out a mutation that's going to lead to breast cancer, Alzheimer's, between, you know, making, you know, green eyes or bigger muscles, right? Most rational people can see a difference. And to me, I don't really see that big a difference in germline stuff as long as you're not enhancing, as long as you're taking out, you know, defects. But that's, you know, I'm open to other people's opinions, obviously. Just my comment.
You bring up a lot of neat points, and one neat thing is in the 70s, when recombinant DNA was first coming online, this got a lot of public attention, and I looked, and there were front page New York Times articles of how this debate is playing out. Right, exactly. So that's the other thing too, is the scientific community got together at the Asilomar conference, which was a meeting in Monterey, where they came up with ethical policies of how to do recombinant DNA safely and what regulations need to be put in place, and the NIH adopted those regulations. What they came up with then doesn't apply as well now because so much time has passed, and I feel like the climate is a bit different because it's not academic scientists anymore who are pushing this. A lot of this is coming from the corporate side where there isn't a process of peer review. So a lot of this stuff is happening more in the dark. So I think it's a really neat example because in the 70s, the scientific community, including the people leading these advances, came together and figured out how to come up with a safe way of doing it. I don't see a similar push happening now, but I think that's the really natural group to mobilize. If we can convince the people doing the research to try to come up with how to regulate it, they might see that as being to their advantage, and it might benefit us all as well.
Suzanne McCauley, great talk. The All of Us program has launched in the United States and is actively recruiting at Northwestern for anyone who's interested. One of the unique things about this genetic and phenotypic discovery program is that it will engage the participants in designing research questions. You've just given us a lot of information about potential benefits and harms. How do we educate the public participants to a level where they can be activated to decide how their data are used?
That's a really good question. I don't have a perfect answer. I have a partial answer, which is if I go back here, I don't know if I can find it, but I talked about that group iGEM, which is a science fair for high school students and middle school students. I don't know terribly much about it. It was developed after I went to high school and middle school, but that actually seems like a way to engage the newest generation with these technologies, and they seem to do it in a way that emphasizes safety and coming up with socially responsible research questions. So I feel like outreach projects, whether I'm not familiar with yours, but perhaps yours, or this iGEM science fair, this is a way of getting people interested and excited about these technologies in a responsible way, and that's useful.
I'm going to sign you up to talk to some high school students.
I just wanted to address Dr. Katz's comment, because I think it's nice to think that we would all have the same views on what is a defect or not, but I know that parents of patients with trisomy 21 might feel very differently than the medical community on whether that's a defect or their life has been altered in a negative way for having their child with that. So I think it's a little bit less black and white than it seems.
One other example I'd bring up is glasses aren't really a natural thing. It's an artificial enhancement to improve vision. It's not really, it's hard to say that baseball players can't use steroids, but people can wear glasses. It's just really hard to pin these things down, even among ethicists.
Well, great talk. In the real world of pediatrics, disease prevention is really important. So you're in practice, mom comes into your office and says, I'm pregnant. I'm a carrier of disease X, and her husband is too. What can we do to prevent this disease X in the fetus in an early stage of pregnancy, if it's even possible?
Help me a little bit. You're wondering, can we use this technology to make it so that parents who are carriers of genetic diseases don't pass those on to their children?
Or once they're pregnant, yeah.
Once they're pregnant. So the technology does not exist right now really to edit the genome, the germline cells of someone who is not an embryo. But that could change quickly, and that's really a next frontier of these technologies. And you make a good point, which is if people know that they're carriers of genetic mutations and they go through the IVF process, there already is a way to pluck out the fertilized eggs that carry the mutations that we're trying to screen out. So it's not a huge jump in the current technologies we have as far as making it so that known carriers can make sure that their babies through IVF don't have the genetic disease, but it wouldn't surprise, this may happen soon. Already we're having luck at editing the genomes of somatic cells, and it might be able to do that so that it's all cells in the body instead of just the somatic ones. Yes?
Yeah, and that was phenomenal. My question is, while I bet a lot of people here could imagine a regulatory environment that attempts to restrict use of these technologies to what we somehow agree are socially beneficial uses, and we might be able to come up with a regulatory environment that decreases the likelihood of unanticipated consequences. But what do you do about bad actors? As you point out, this technology is widely available to people who want to access it, whether they're in an NIH-funded institution or in their basement.
Yeah, you ask a good question, and
Thank you.
I don't know that people have good we need to come up with an answer, but we don't have a great one. We have partial answers, so there's a voluntary scheme by some of the companies that supply mail-by-order DNA where if they are too similar to known pathogens like smallpox, say, then they're not allowed to send it out. But these are really easy to circumvent. And also, you can potentially edit the genome at home using these different technologies. So I don't know, and I hope it's not going to take a really bad event for us to work on this with more heft. To me, this seems like a much easier technology to use if you're a bad actor than the other ones that are around, and that makes me really worried.
Dan, that was awesome. As a fellow trainee, I'm really proud of you that you're stepping up and delving into something that we don't get formal training on.
Until now.
Yeah, no, right. That's absolutely right. And actually, that's the point, is we're all of us, those of us in the room right now, are going to graduate quickly. Like, years fly by, and we're all working in the clinic, and but the time that you took to really delve deeply into this topic is your time, and it's energy that you put into it, and it's out of an interest that you have. But it's knowledge that we are for the most part going to require. We're going to be required to have to be the gatekeepers, or to be advisors, or to even give any kind of advice in the clinic. And one of the things that I'm personally struggling with, and I know a lot of my co-fellows around the hospital struggle with, is we start sending tests, or we start delving into treatment ideas that even those of us that are interested in it, it takes more time than we have to really understand at a level that we can advise on. And really, my question is, do you see a pathway from your experience at Lurie or from your experience in med school? Do you see a pathway where this kind of knowledge can be more easily integrated? Because as far as I can tell, most people stop thinking about genetics at any kind of formal level after Genetics 101. And then it kind of pops up like this. You get excited about it again, but you realize so much has happened since the last time I thought about this. And the barrier to entry sort of seems like it's missed.
Yeah. So let me build on something you brought up first, which is I've delved into this on my own time, and I want to say that's really a credit to this residency program, that I was able to do a program on the East Coast in my spare time, and that took administrative help to have my schedule rearranged and things like that. So I do appreciate that the program here made it possible to branch out into things beyond just inpatient medicine. So thank you. As far as how do we get more people interested, I think the common press is a good place to start, and there have been some really good articles out there. I mean, I've cited a lot of New York Times articles here. The New Yorker had a wonderful article a few months ago about these technologies. All the scientific magazines do. So I feel like that's a place to start, where if we can get people in the press who get it and understand these issues and are able to write good articles about that, that will help a lot. And then I don't know how to make it so that this is more part of a medical curriculum. I don't even know how important I don't know how much it's part of a doctor's job to be working on these broader issues.
It's a pretty big deal in neurology.
Yeah.
I can only speak for what I do, but it's a big deal.
Yeah, but I think these sorts of things are great, that there are opportunities to share the research that everyone's working on.
I think we have to stop now in the interest of time. Again, a final thank you to Dan.